demonstration of whole cell patch clamping technique is critical, because there are many steps that require precision, proper electronic compensation, and practice for generating usable data.
After the desired incubation time has passed, place the cells on the stage of an inverted fluorescence microscope to determine the transfection efficiency. Compare fluorescent and DIC images to determine the percentage of cells transfected. Return the culture dish back to the incubator. Clean 12mm glass cover slips and store in ethanol until use. Remove the residual ethanol by flaming in the sterile cabinet. Place the cover slips in a 24-well plate and coat them with 0.2 mg per milliliter poly-D-lysine. 24 hours after transfection, trypsinize the cells and plate 4 × 10⁻³ cells per well in 4 to 8 wells of a 24-well dish containing poly-D-lysine coated glass cover slips. Incubate for an additional 24 to 48 hours. Prior to performing whole cell patch clamping, prepare patch electrodes using a 2-step vertical puller and borosilicate electrode glass. Ideally, the finished electrode will have 3 to 7 megaohms resistance and will not require additional manipulation, such as heat polishing. For patch clamping, place a cover slip with transfected cells in a 35mm dish containing 20 mmol of potassium or 20 K external solution. Place the dish on the stage of an inverted fluorescence microscope. The patch clamping setup used here employs a perfusion system. This system contains the extracellular solutions that will be applied to the cells, which are gravity fed through tubes connected to a manifold. Each solution is controlled remotely with a solenoid pinch valve. Use DIC to focus and find cells. Switch to the YFP filter to locate the YFP positive cells. Once the YFP positive cells have been identified, position the tip of the perfusion system near a fluorescent cell, 20 to 30 microns or 2 to 3 cell lengths away. Fill an electrode with internal electrode solution using a syringe and flexible needle. Gently tap the filled electrode to remove air bubbles from the tip. Attach the electrodes to the head stage of an AXO-Patch 200B amplifier. Check that the settings on the amplifier are gain equals 1, configuration whole cell switch is beta equals 1, and the low pass Bessel filter is set to 10 kHz. Apply positive pressure with the syringe and clamp to keep the electrode tip clean. Then use a micro manipulator to place the tip of the electrode in the bath and move to above the cell. Switch the amplifier to V-track mode and the meter knob to V-track. Then use the pipette offset potentiometer on the amplifier to zero the pipette potential by bringing voltage on the amplifier meter display to 0.00. Alternatively, switch to V-clamp mode on the amplifier and select the test pulse button to deliver a test voltage step. The rectangular voltage step will appear in the seal test window of ClampX. Adjust the pipette offset on the amplifier to bring the baseline current to 0 nanoamperes. The seal test will show the electrode resistance, which should be 3 to 7 megaohms. Next, close the seal test window. Select Acquire, Open Protocol in the drop-down menu. Then select a saved voltage protocol. Here the protocol selected has been pre-programmed to deliver a single voltage step to -100 mV and ramp to +40 mV every 2 seconds. This ramp protocol allows for observation of the reversal potential and for inward rectification property of Gerke channels. For voltage-gated ion channels, a rectangular voltage step may be more appropriate. Using the fine adjustment of the micro manipulator, approach the cell. Release the positive pressure and after touching the cell surface, apply slight negative pressure. Monitor the resulting increase in resistance in seal test. Once resistance is in the gigaohm range, the electrode has sealed onto the membrane. This is referred to as a gigaseal. Using a syringe, apply a pulse of negative pressure to break the membrane and gain access into the cell. Once in the whole cell configuration, the seal test will register large capacitive currents that are visible as spikes or transients at the beginning and end of the test pulse in seal test. Next, click on the membrane test button in ClampX. Record the access resistance, membrane resistance, and membrane capacitance in a notebook. Verify exponential fit by eye. The membrane capacitance is proportional to the cell size and will be used to calculate current density. Access resistance and membrane resistance are used to evaluate whether the recording will be satisfactory for continuing. A good recording will have a low access resistance. Now, close the membrane test window and click on the seal test button again. In the seal test window, set V out to -40 mV. To compensate for membrane capacitance and series resistance on the amplifier, switch the whole cell cap on. Adjust the whole cell cap and series resistance back and forth until the transient is minimized and the current is a flat line. If needed, adjust the pipette capacitance with the fast cap knob on the amplifier. Too much or too little compensation will leave a residual capacity transient. Next, turn the percentage prediction knob to 60 to 80%. Readjust the whole cell cap, series resistance, and fast cap as needed to obtain a flat line in the seal test. Finally, turn percentage comp to about 100% without oscillating the cell, whilst trying to keep the pulse as linear as possible by readjusting whole cell cap, series resistance, and fast cap potentiometers. Once resistance and capacitance compensation is achieved, write down the values set for the amplifier, capacitance, series resistance, percent compensation, percent prediction, and lag time in a notebook. Turn on the external solution valve for the control 20K bath solution. Set low pass Bessel filter on the amplifier to 2 kHz. Then, start recording currents by clicking on the record button in the software. Look for the inward rectification property of the Gerk current. This is seen as a large inward current, negative to the potassium reversal potential that is determined by the Nernst equation, EK, and a small outward current positive to EK. After recording a stable baseline, determine receptor-mediated activation of Gerk currents by applying extracellular solution 20K plus carbachol. Once peak activation is observed and current reaches steady state, switch back to the 20K bath solution and wait until baseline current level is reached. Next, to study alcohol activation of Gerk currents, switch to external solution 20K plus 100 millimolar ethanol and wait for peak response. Then, switch back to the 20K bath solution and wait until baseline current level is reached. Finally, to determine the basal Gerk currents, switch to the 20K plus 20K barium solution and wait for the response. Here, the inhibition of Gerk current should be evident, being most pronounced at -100 mV. Throughout the procedure, note the trace sweep number corresponding to the application of different extracellular solutions. A single clamp fit file will contain all the sweeps for the entire experiment. To analyze Gerk data, use the clamp fit 8.2 software according to the instructions in the accompanying written protocol. Two different whole cell recordings were obtained from HEC293 T cells expressing either wild-type or mutant Gerk channels and analyzed according to the instructions in this video. As shown here for wild-type Gerk2, the panel on the left contains a window with several superimposed currents that were produced by the voltage ramp protocol in the presence of different solutions. Note the inward rectification. That is, the larger inward current at -100 mV around 50 milliseconds on the current trace. The small outward current between 0 and +40 mV around 200 to 260 milliseconds on the current trace, originates from an endogenous voltage-gated potassium channel. It is not the Gerk current. Cursor 1 is placed over the maximal inward current and the cursor values are written to the spreadsheet. In the spreadsheet, the trace number and current at -100 mV are seen. The data include basal current, carbachol-induced and alcohol-evoked currents, as well as a barium-inhibited current. Likewise, the voltage ramp currents in the presence of different solutions, 20K plus 100 millimolar ethanol, or 20K plus 5 millimolar carbachol, or 20K plus barium, are shown for the mutant channel L257W. Note the significantly smaller alcohol and carbachol-induced currents. These findings suggest that this residue plays an important role in gating of Gerk channels by alcohol and G-beta gamma subunits.
After watching this video, you should have a good understanding of how to construct a mutation in an ion channel and study its effect on channel function using advanced electrophysiology techniques.
Once mastered, this site-directed mutagenesis technique can be done in 2 to 3 days and scaled up to 5 or more mutations.
The development of these techniques paved the way for ion channel biophysicists to explore basic properties of ion channel function and gating. In addition, they allow researchers to study the human diseases that are caused by a single mutation in an ion channel gene.